Hello, everyone. Good morning. Welcome back to our seminar series on Translational Biomedical Engineering. It's my great pleasure to host Professor Jared Churko from Arizona, University of Arizona with us today. He will be talking about engineering induced pluripotent stem cell-derived cardiac organoids for arrhythmogenic cardiomyopathy modeling. Before we start, please, as usual, please use the chat box to communicate your feelings and then your thoughts with others. But if you have any questions, please use the Ask a Question box, which is in the bottom of the screen. Also, our next speaker is Professor Thomas or Thomas Jarvet from Polytechnic Montreal. He is also a co-founder and CTO at Mesochip, and he will be talking about his new advances in this development of these microfluidic devices for various applications. If you would like to receive most up-to-date information about our e-seminar series, make sure that you follow us on Twitter. Our handle is TransLBME. You can also always follow me and Human, co-organizers of these e-seminar series. On Twitter and LinkedIn, we make announcements about future upcoming talks on these two platforms as well. Last but not least, I'd like to thank Montreal Transmedtech Institute for sponsoring this event since almost the beginning of this e-seminar series, which was 2 years ago. Thank you, Montreal Transmedtech, for supporting this event. So with that, I'd like to introduce our speaker, Professor Jared Churko, who received his PhD from Western University in Canada, where he studied the role of connexin 43 in skin regeneration and other diseases. He then joined the group of Dr. Joseph Wu at Stanford University, and I think he was one of our speakers as well. And in this event in Cardiovascular Institute, where he developed a protocol to generate human-induced pluripotent stem cells using a non-integrating virus under defined conditions. And then they compared the transcriptomic and epigenetic differences between these iPSCs and derived and human embryonic stem cell derived cells, and studied the gene expression profiles and transcription factor regulators involved in iPSC-derived cardiomyocyte heterogeneity. He became an independent investigator at the University of Arizona in 2018 after receiving the prestigious NIH K99 Pathway to Independence Award, which is highly competitive. And he's currently the director of University of Arizona Human-Induced Pluripotent Stem Cell Core. His research group within the Sarver Heart Center utilizes iPSC-derived from patients with heart diseases to study patient-centric disease mechanisms, and his lab exploits gene editing technologies and bioinformatics to understand the fundamental molecular mechanisms leading to heart diseases. His lab engineers differentiated cardiac cell types into 3D tissues to better model heart disease pathophysiology, and employs various imaging and biochemical assays to interrogate the functional changes involved in heart diseases. So with that, I'd like to invite Professor Churko to share his slide, and then the virtual floor is now yours, Jared.
No. Can you see this or no?
Not yet. Perfect. Yes.
Perfect. Well, thank you so much for that introduction. I hope everyone can hear me. As the title kind of states, we're interested in a disease called arrhythmogenic cardiomyopathy, And it's one of those really difficult type of heart diseases to model, and I'll explain why. So just a quick kind of overview of our lab, because I think that's always good to kind of see where I'm coming from. But our lab really does want to look at the patient themselves. So we find cohorts, we have collaborations with different physicians at the U of A, where we have patient cohorts that have all different types of heart disease. And we take blood samples from those patients because it's a very easy, simple thing to do. And then we generate iPSCs. So I think a lot of people in the audience will know about iPSCs, a lot of these talks, but it's really a cell type that has the potential to go into any single cell type in the body. And one of the great kind of advancements is being able to gene edit these cells very robustly, and we use CRISPR-Cas9 to do that, so that we have isogenic control lines that can go along with these cells. From here, we differentiate both our isogenic control lines as well as our patient lines that maybe have different mutations causing the heart disease into multiple different cell types within the heart. So I know a lot of laboratories are focused just on the cardiomyocytes because those are very difficult to derive cell type. You can't just take these cardiomyocytes from patients. So to have the ability to generate cardiomyocytes in a dish from these patients and model different diseases is extremely powerful. But we also focus on different cell types, and I'm going to maybe only touch about one of them in this talk, about how we're using epicardial cells to also model arrhythmogenic cardiomyopathy. So from here, we assemble these different cell types into bioengineered models, which will be the majority of the talk that we focused on, based on the I guess the goals of this seminar series. It's not going to be data heavy. I want it to be a little more light in terms of the methods that we're employing to actually study, and kind of a lot of the caveats that we had to face in order to be able to build these kind of tools within our lab. We're not just stuck on the bioengineered models. Of course, there are some times when there's just structures that we're still not able to generate in the dish. So we do have mouse models of the disease. But our lab primarily focuses on doing high-throughput sequencing, different types of proteomics, mass spec, to really get more of a global picture of what are the different disease mechanisms, how is a certain protein interacting with another one, and how does that mutation disrupt this interaction to cause downstream signaling cascades involved in the disease. And then we also employ a lot of functional and physiological assays to validate some of these changes that we see in the dish. So as the title kind of states, we're looking at this one disease called arrhythmogenic cardiomyopathy. And this is a perfect disease for utilizing all the different bioengineering tools out there. And one of the kind of, I'll just explain the disease in itself. It was previously called arrhythmogenic right ventricular cardiomyopathy. And that's very important because somehow unique to the right ventricle, there was most of the pathology. So if we're trying to bioengineer a system where we can model this, we need a system where we can model more right ventricular cardiomyocytes versus the left ventricular cardiomyocytes. So this disease is also a progressive disease where initially there's no different symptoms, the heart seems fine, but over time there's different changes within the heart, and which could be deadly. The frequency of the disease is 1 to 1,200 to 1 to 5,000, but this frequency really depends on the population itself, which I'll explain in another slide. And 35 to 50% of these patients have no identifiable associated variants. And this is also something I think really great about iPS cells, is that you might not necessarily need to know the mutation that the patient themselves harbor, but you could use their cells and their iPS cells to still model the disease because you know that those patients have the disease. But the key, I guess, clinical characteristics of the disease is that there's cardiomyocyte loss, there's fatty fibril infiltration. So if you see here, here's a normal heart on the, I don't know if you guys can see my cursor, but there's on the normal heart, there's no fat infiltration in the myocardium. But on the right, this is a regenerative cardiomyopathy. You see these fat infiltrating into the heart itself. So this is a very unique feature of this disease. So it would be really cool if we could model what's going on with this fat infiltration and how different mutations are leading to this pathophysiology. And the last two are also really important. They're arrhythmias. So if you have an abnormal heartbeat and your heart is not functioning properly, this could lead to sudden cardiac death. So it is a very serious condition. And I should just say, like, we really wanted to look at the clinical profile of disease and see how in a dish can we model these different aspects. And I think that was really the way we're trying to make this more translational, is like see the current tools that we have and how can we take it to the next level to model specifically this disease. So as I said, this is a disease that progresses over time. So there's this concealed phase where there's no overt abnormalities, and then there's this electrical phase that occurs where you can start to pick up different electrical abnormalities, but then finally it gets to the structural phase where you get this little black spot here, this cardiac MRI showing that there's fat and fibrosis within the myocardium. And once you reach the structural phase, it's very difficult to go back. So we want to be able to identify the mechanisms that's leading to the earlier phases, and iPS cells are great for this because you can now look at an earlier cell type over time to see how disease mechanisms can manifest. So I also mentioned this is very important for sudden cardiac death. In looking at a study of all sudden cardiac deaths from the National Collegiate Athletic Association, they found that 16% of these sudden deaths were due to cardiac-related events, and then when you break those events into what type of cardiac event, 3% in the U.S. was due to ACM, so arrhythmogenic cardiomyopathy. However, in the same sort of, assay, in looking in Italy, where mutations causing ACM are actually more prevalent, we do see about 22% of sudden deaths, cardiac deaths, are due to ACM. So, what are the different mutations that have been known to cause this disease? They are primarily involved in the desmosomes. So there's listing of plakophilin, desmoplakin, desmoglin, desmocolin, plakoglobin. But it's not just desmosomes that can relate to this. There's other types of proteins that have been mutated to also cause the same disease phenotype. So there's adherence junctions, desmins, titin, and ions as well. So really trying to find the link between all these different mutations and how it causes the same downstream disease is one of the goals of our lab. So what is the desmosome? I think this is just basic cell biology, but the desmosome is composed of multiple different proteins, and it's really, there's these desmocolins, desmoglians that act kind of like adherence junctions to be able to attach one cell to another. And those are linked to plakophilin, plakoglobin, desmoplakin, and eventually it's linked to desmins, which is this intermediate filament within your cardiomyocytes. So if we take a look over here, this is what the desmosome looks like. And these desmosomes in the heart are actually localized at the intercalated disc. So the intercalated disc is when you have two cardiomyocytes joining each other. At that interface is something called the intercalated disc. And that's where a lot of these junctional kind of complexes are, as well as a lot of the channels that allow two cardiomyocytes to connect and to communicate together, because this is eventually one of the main ways that you have synchrony within the heart, so that you constantly have a beat rate that is synchronous and doesn't cause arrhythmias. So this is a very important structure. If you can imagine, you have a heart that's constantly beating nonstop all day. You have to have a way for two cells to maintain their junctions, and desmosomes are one of those, where they're kind of these strong fasteners that button two cells together. And just a quick review of the different research models that are currently out there. There's actually a zebrafish out there that has been reported to be a model for arrhythmogenic cardiomyopathy. The fish have two chambers. They have an atria and a ventricle, so it's not necessarily that they have a left or a right ventricle. Obviously, abundant rodent models are out there, and interestingly, actually boxers can develop naturally arrhythmogenic cardiomyopathy. So that's also a model. But we're really interested, obviously, in the clinical findings and the human condition. So in order to study arrhythmogenic patients, most of this is going to be clinical findings. So more imaging-based modalities that there's going to be non-invasive. We can do epidemiological different findings. Sometimes you can get tissue during OR, but that's very difficult. Human tissue is, at least for me, is very difficult to obtain. But we've taken the approach of an iPS cell kind of route in order to study this disease. So most of you already know that iPS cells have replaced human embryonic stem cells, which do pretty much the exact same thing. This was the Nobel Prize by Yamanaka back in 2012, where they identified 4 different factors, which you can take any somatic cell, add these 4 transcription factors, create iPS cells, and then from there we can differentiate these into every single cell in the body. So we do this a lot in our institute right now, where I'm directing this iPS cell core, where a lot of our patients are happy to donate just 4 mils of blood. We isolate the PBMC layer, so we don't have to do any scars, which used to happen when you had to take fibroblasts with punch biopsies. And then we're trying to actually advance this a little further by taking smaller and smaller amounts. So if you could just have a finger prick, I think that would be a little bit more amenable to taking blood for some of these patients and be able to only take those cells and reprogram them, which would be even an easier kind of method. So, so do we have any iPS cells from patients that have this disease? Yes. This was in collaboration with Hugh Calkins at Johns Hopkins, where he has 2 families that actually had different mutations. And when you sequence these patients, they had mutations in a protein called PKP2 or plakophilin 2. And so just up here, we're showing these 2 mutations that we have. Over here, we did some Sanger sequencing to validate, yes, this leads to a frame shift here. So we see that the sequence is kind of more out of order after this point. But just another kind of fact about PKP2 is that it's actually the most common mutation associated with arrhythmogenic cardiomyopathy. So we have at least 10 lines in the lab right now where we're using to study this disease. We've generated multiple gene corrections on these patients, but not only did we correct the mutation, but we've also introduced the mutation. So we also want to take control samples and then add in mutations to see, is it directly the patient background, or is it something else that's not specific to this patient. So we've made a lot of isogenic lines in these patients, and we differentiate them in parallel so that we can really see differences that are just due to that one single nucleotide difference. So obviously in disease modeling, we have to ensure that we're looking at the right isoform. PKP2 is interesting in that it only has 2 isoforms, but it's still, if it's expressed throughout the body, sometimes you wonder why diseases only affect a certain system, and this could be due to splice variants or different isoforms of the protein being expressed. So we've kind of, the structure of PKP2 is not known really. There's only some of these armadillo repeat regions here that have been resolved. So we modeled kind of the full length of PKP2. We looked at these different repeat regions here, armadillo repeats 1, 2, 3, all the way to 9. And then in order to assess which isoforms expressed in the heart, we looked at our RNA-seq data. And our RNA-seq data, if you guys are familiar with that, you can look at reads that map to different junctions. So on the bottom here, we have the 2 isoforms, and then these larger kind of boxes are the exons. And then here's the introns, which are just this thin line. So you can count the amount of reads that actually overlap between this exon and that exon. And then you could do a ratio comparison to see, is there equal numbers and are there different splice variants based on the amount of exons? So theoretically, you'd expect there to be almost equal amounts if there's all the same isoform. And you can look here, you can see in this isoform of PKP2, there's this exon, but in this isoform, there's not. So this is isoform A, which is a shorter. In the heart, there's actually only 6 reads that overlap with this. So the predominant isoform in the heart is actually this shorter isoform, PKP2A. So this is very important for us, at least in our, when we're overexpressing this, or we're doing a lot of our biochemical assays to be able to see what specifically is different with this isoform versus other isoforms. So this is just also a schematic showing that our mutations are 2 different C-terminal truncations. So one actually leads to a frame shift, a couple of amino acids, and then a stop, whereas this different PKP2 isoform or mutation in another family cohort actually leads to a stop codon. So we're really missing the C-terminal tail of PKP2. So I'm sure a lot of you guys have seen the different assays out there or the different methods to create cardiomyocytes in a dish. So the field has definitely been now for a long time, maybe at least within 5 years, to a point where you could take iPS cells and you could differentiate them into 90 to 100% pure cardiomyocyte populations. This is also very important for disease modeling. Sometimes you want different cell types in there, but usually if you want to study the mutation only in a certain subset of cells, you want very pure populations, and you want it to be consistent between different lines, because you don't want to compare an unpure population with a pure population, and then so your assay might be actually telling you something that's not really true about the disease, but it's true just based on the differentiation potential. So the field really moved to a point where you can, even with the naked eye, see very nice beating waves of propagations of cardiomyocytes in a dish. And it's become a lot cheaper, where now you really only need 2 small molecules to do this. There's no more very expensive proteins involved in this. So we could take one called CHEER, and that activates Wnt at this time point, and then we inhibit Wnt with IWR1, and over time you get beating cardiomyocytes. So it's really amazing. But really the question is, what type of cardiomyocytes are we generating? Are we generating atrial, nodal, ventricular? Is it left ventricular? Is it right? And these are all very important things to be able to model different diseases, especially in the heart. If you have an atrial arrhythmia, or if you have something that's dependent on Purkinje fibers, maybe you want to generate that type of cell in a dish. So what type of cells do we, or this cardiomyocytes that we generate from this protocol, what type of cells are they? Well, we've done some single cell RNA sequencing, we've clustered them. Each of these rows is a gene, and then each of these columns is a cell. This was done a long time ago with something called the fluid iPSC-1 system, where you could really only look at 96 cells at a time. So this is about 54 different cells, but we could still really look at the different clusters to see what types of subpopulations of cardiomyocytes are in there. And then we could look at these different populations and say, well, are they more atrial or ventricular? So here, if we look at this population, which expresses NRF2, these all have more of an atrial phenotype, whereas this population has a ventricular type of gene expression profile. So you might think that you're actually creating a mixed population of both atrial and ventricular cardiomyocytes. Well, if you look more at a long-term kind of continuous trajectory analysis of sampling cardiomyocytes at different time points, you can actually determine what's going on with the different populations and are they stable. So are the atrial cells atrial cells? And this is, again, very important for disease modeling, because if you're looking at day 30 cells versus day 60 cells in culture, you're going to get a completely different answer than a cell type that maintains its kind of gene expression profile. So to answer this, we turn to the 10x genomics platform. I'm sure everybody has heard of this, but it's a job-seek method where you can take microfluidics and pump different cells throughout these different microfluidic chambers, capture individual cells within each of these different, droplets, and then perform all your barcoding within that droplet. And then after you sequence it, you can go back and say, all these transcripts were from a single cell, and then we can get very fancy UMAPs or T-SNE plots to look at the heterogeneity of the population. So we looked at 3 different days, and we found that over time, even if you look at day 5 of differentiation, day 14, day 45, here they're colored in different colors. You get different populations of cells. At day 5, you get this population, this population. Day 14, now you're starting to see this population. And day 45, now this one, this one, this one. So really, the beauty of single cell RNA-seq is that you can actually not look at these other cell types that have been generated and really focus in on what you're trying to address. So if we look at the transcription factors within these different populations, we can see that at earlier stages, there's the expression of more of the progenitor cells. So here's OCT4, SOX2. Those are the reprogramming factors. Still, even at day 5, we see these cell types. So at least in the clinic, if you want to inject these types of cells, you want to make sure that they're after day 5, because possibly these cell types are still in a progenitor state, can cause teratomas. So, but if we look at just the cardiomyocyte population, we start to see that they express factors such as HAN1, F2C, and YH6, and then late stages, so day 45, they start to express this NYL2, which is a ventricular specific marker. So over time, these atrial type of genes actually decrease, but the ventricular type of markers increase over time. So if you generate iPS-derived cardiomyocytes using this protocol, chances are these are going to be ventricular type of cell. And to answer, is this more of a right ventricular type of cell, which we might want for arrhythmogenic right ventricular cardiomyopathy, or left? Well, they express this HAN1 transcription factor, which is more involved in the left ventricle, but really the left and right, if you look at the gene expression profiles in the human heart tissue, they're almost identical. It's actually amazing. So let's differentiate our iPS-derived cardiomyocytes into, like from the patients into cardiomyocytes. And we see that if we look at the EM, we do see disruption in the desmosome, so the architecture is really disrupted. And what's interesting is that PKP2, which is that protein that we're looking at, is still expressed. So it seems like the patients still have a lot of PKP2 expression. It's a little disrupted, but if we look at connexin 43, which is one of those channels that dictate cells to communicate intracellularly, that in the control cells we see the nice punctate expression profiles, but in our mutant we actually see more of the diffuse type of profile, which could indicate that these arrhythmias are due to somehow dysregulation of connexin 43 communication between the cells. But this is all monolayer. We really wanted to look at heart tissue itself, and this is primarily because the heart does need a 3D interaction. They need to communicate more unidirectionally so that the heart synchronizes in one direction so you don't have arrhythmias. Whereas in the monolayer system, you primarily get cells that are going to be having connexins around their entire cell border, which doesn't really help if you're looking at arrhythmia type of analysis. But the way we approached this, and this was like 3 or 4 years ago, was this engineered heart tissue system. And this was pretty much just released back then. So we thought this is a great platform where we could start to develop these different organoids in a system that's commercially available, and then fine-tune the system for what our experiments needed. So this is, I think actually the EHT Technologies was now bought out, so a lot of this equipment is not available. But the way this platform works is that you take agarose, you put in these Teflon spacers, which would form this hole after it hardened. You take your you take a cardiomyocyte mixed with different matrigels or matrices into this hole, and then you place these the silicone rack, which have these 2 different posts here, and so the cardiomyocytes and the cellular mixture would harden between these 2 different posts, and then when you could remove the silicone rack, and then you could place in the media, so you have the suspension culture of 2 cardiomyocytes, and then as they contracted, you can actually measure the force that the cardiomyocytes contracted, because we knew the Young's modulus of the post itself. So this machine was available in one of the other labs, but we didn't buy our own because it was $100,000, and starting a lab we had to really be mindful of what type of technology we're wanting to invest in. So we wanted to take one of our microscopes that we had in the lab and see if we could generate similar sorts of results using this platform, but without this this camera system that's had very expensive software. So one of the students was working on an open source method in order to take these contractions and look at take the videos of the contractions and look at how these contractions changed over time. And so we started out with one thing was remodeling the platform itself, and starting a new lab, and we're trying to train students. I think a lot of us see that there constantly contamination issues. So if these cells take two months to generate, or at least 2 weeks to get cardiomyocytes, then you assemble them into the EHT format, and then you're trying to image them, you're trying to paste them, and they get contaminated. And one of the things that we realized was that it was probably due to the lid. It wasn't really fitting on top of these electrodes here. So one of the students decided to just reprint a lid and then have just a glass insert in there so it can be washed, ethanol sterilized, and then remove a lot of these electrodes that were sticking out to just 2 wires, so that we have a system that's enclosed that we can transfer to and from the microscope without actually causing any kind of contamination. So that was one of the first things we did, and we also added in a spacer, because if we want to stay consistent between different runs and just have a script to look at different XY coordinates on our plate and video that. All we'd have to do is have this spacer to hold them into place so it's consistently the hESCs are always going to be in the same spot every time we run our and this is just some really basic, 50 cents to 3D print, but it saved us a lot of money in failed experiments, contamination, or time lost. And one of the big things that I was kind of shocked at is how expensive the pacers are. So this is a $5,000 apparatus that we bought, which is great. Like, it works amazingly, but you can only really do one row of hESCs at a time. So we weren't going to buy 4 or 5 to do multiple cell lines, multiple different experiments, looking at different pacing schedules. And we really couldn't afford just actually waiting for a complete experiment to be done to restart the next one. So one of the students decided that this is something easy that we could do in the lab. And so we took an Arduino project where we've added in different, the ability to actually pace our hESCs just using a $100 type of project. So if anyone's interested in just building this out in their lab and saving the lab some money, and if you're really into tinkering with these types of projects, then let us know and we'll send you the schematics for it. But we definitely wanted the ability to adjust the frequency of these of the pacing itself, because as like I mentioned, these cells need to be at a certain level to be able to so that you can study differences in how the heart is actually functioning. And so a frequency of 1 to 4 hertz is perfect for human cells, because even at 3 hertz, that's 180 beats per minute, which is more of an exercise profile. And one of the reasons we wanted this type of system is to make sure that we can look at maybe more exercise conditions. So one of the kind of aims of my grant was to look at a model of cardiac stress to be able to cause ACM. So just in the dish, just beating cardiomyocytes, maybe this wasn't enough, that we needed to actually pace them to a level that the human heart experiences, and maybe we can answer some of the questions of the disease mechanisms of why there are sudden cardiac deaths in a lot of these athletes. But just as a side, like this aim got killed because they really hated the term exercise for cell modeling. So we've now kind of toned it down and saying it's more stress, but I do think that this is still an engineering approach that is addressing a really clinical or fundamental question of, does cardiac stress or pacing actually, is that necessary to be able to reveal the phenotype in our patients themselves? So this is a model we've been looking at, and we've been pacing them 3 hertz to be able to now look biochemically of, is there a difference between and cells that have been paced versus not, and does this cause more of a disease phenotype, and can we now start to see arrhythmias during the pacing? And one of the, I guess, it started out as a side project, thinking it wouldn't take a long time, but it's kind of been morphed into more of a monster at this point. But because we didn't buy this 20,000 piece of software to be able to analyze our contractions, and we looked through the literature and we found maybe one software that was available to do this, we decided to take it on ourselves to develop an open source platform to be able to analyze our engineered heart tissues. So here you can see there's a beating, one of the posts is beating, contracting. So we wanted to take the displacement of this and translate this into contractions. And we wanted this to be, very user-friendly. I think the major thing in developing software is that it's got to be, you click on something and it gives you a result, and you have to be able to understand it. Can't give errors. It can't be very user-specific. Whereas a lot of the times you'd be like, well, if I change this little part in the code, it works for these figures and it doesn't work for these. So it has to be very universal for all different types of engineered types of tissues, organoids. And we've even made a monolayer version of this. So if we just like contractions within a monolayer, we can look at the differences and calculate at least the force of contraction. And so this was just like one of the things that we really wanted to design was making sure that the user could put the right types of data in there, and even something as simple as different frame rates, you can get different results, or if they had MP4s versus AVIs. So a lot of that had to be incorporated in the code, so it's very user friendly, and the user itself doesn't have to think about any of these caveats, but we did so that we can make sure that the results are consistent and you're going to get constant data out of this. And then another kind of thing which I didn't really appreciate was the algorithms involved in this. And we've had a few different people kind of take a look at this because there are different ways that you can analyze contraction data. And one of them was just to look at, you take all these pixels and you make your construct black, and then you look at white area around it, and then you could look at just how many white pixels are there versus black pixels. That's a very computational, less intensive way to do that. And then there's, we're working, actually just go to the next one. We're working with two PIs here in electrical and computer engineering that developed an algorithm called anomaly detection. So it'll take groups or chunks of data at a time. It'll divide your trace, all the data into these chunks, and it'll look at where these chunks actually change, and that can be an indication of where the start of a peak is versus the end of the peak is. So a lot of work went into this, and a lot of very smart students, especially high school students that have worked on this. And this was something that was perfect for the pandemic. We had two high school students that could sit at home. Nobody can come into the lab, so they would work on this for their senior projects, and they really enjoyed it because there's just some really, really smart students out there now that love these types of projects. So we're able to incorporate a lot of their data into this, but this has been a lot of different people have kind of had their hands on this, and it's interesting just in software development how one person touches one thing and then the rest of it breaks, but that student's already now UCLA, so it's constantly a learning curve, but we're trying to get it to a point where it's something that we could release as a very robust system that is very user-friendly and can give you good data. So just a little bit of kind of features that we've been trying to utilize, and it's auto detection of the peaks. So using an algorithm just to detect the different peaks, because a lot of this data is not as clean as I'm showing here. A lot of times there's noise, even if there's some frequency in the background. If you're running a pump, you get vibrations, and then your contraction analysis doesn't look as clean. So we have some filtering processes in there as well. And then it can classify the peaks based on shape, based on the height. So height would be how forceful the contraction is. The duration can tell you different things, such as relaxation, or even differences in different ion channels. So we've tried to really take the peak itself and break it down and to look at all the different aspects, because even just slight differences can tell you the difference between an abnormal contraction versus not, and computers are way better at detecting this than we are. So we also have the ability to just import all your files at once and do a batch analysis, and so you see all the data at once. So I think I spent too much on this, but we'll just get to more of the data. So pacing these cells these EHTs actually increases the force contraction. So it's one of those things that is very important, because you do want to look at more mature cells, but again, you have to make sure every equal across the board between looking at ACM versus not cells, to be able to really say, is this more maybe a more mature profile, or is it a disease profile difference? So we validated this looking at different methods, but over time, and we looked at different frequencies. And just one thing, if you guys are interested in getting into this, you can't just take these iPS cardiomyocytes and case them at 2 hertz. You have to work them up. So it's almost like you do have to condition them through the different frequencies before you can move on to the next. So it does take another 20 days after differentiation. Very long experiments. We've looked at ACM versus control unpaced conditions. There doesn't seem to be any significant differences in the force generated, but there does. I'm not showing you the pace data right now, but the pace data, I can just tell you that at higher hertz, the cells don't seem to respond as well. So they, and we're trying to figure out what that is at the moment. So, but one thing that's great about this is that not only can you see when things contract, but you can look at differences between contractions. So if you have a lot of contractions at one time frame and not a lot, so a lag period, this can indicate arrhythmias. But we wanted maybe a more sophisticated method to looking at these arrhythmias. So we've looked at 2 different dyes, because electrophysiology work is, patch clamp is very laborious. So we've implemented a method where we can just image ourselves using dyes to be able to calculate the contraction or the traces themselves, and then also import those traces into our software so that we can look at differences between even calcium. So if you have a longer kind of profile, you can get increase in calcium handling capabilities. But what we really wanted to do was look at simultaneous voltage and calcium-based imaging. And so what we needed for this was a very high frame rate camera, because you needed to capture on the way up every single frame of the intensity of this different dye and on the way down. So we've developed a system now where we can look at both calcium and voltage at the same time, or we can look at voltage differences, and this is a ratiometric dye, which is Fura 2. So you actually look at 3 different wavelengths here. We're looking at 340, 380, 470, and we're rapidly changing between these different these different filters, and eat at about 50 frames per second. So this is a very fast, sophisticated type of analysis we can do. And then when we take our control cells, we can see that there's very consistent, robust beating, like there's no variability between the different peaks. And then when we look at our ACM line, we do see arrhythmia types of formations using our platform. So this is a good state that we're at, where we can test different drugs to see if there's ways that we can actually mitigate these arrhythmogenic events, and maybe what's even causing them. So, as I said, in order to make these our results more translatable, we did need to incorporate some mouse models into this. So we developed 2 different mice. One has wild type full length with another protein that we've attached to it, and then also a mutant mouse, in order to see what are the different cellular profiles. So always tissue engineering. If you're just looking at the cardiomyocytes, that might not be the best situation, because maybe the disease is in the endothelial cells. And just as a reminder of the disease itself, there's this fat fibril infiltration that gets into the heart, and there's fibroblasts that proliferate. So we want to look at differences in fibroblasts. Maybe it was, and can we look at the human iPS cells and do the same thing? So, but what's unique about this disease is that the mice actually don't develop the same structural differences that the humans do. So this is very important. Why we're using iPS cells is that they don't develop this fatty fibril infiltration. their heart rate is beating at rest at 500 beats per minute. So just think about trying to analyze arrhythmias when it's already at 500 beats per minute. So we are using our mice to look at more of the different cell types that are probably causing the disease, some of the pathways that are doing so, and then we're translating this into our iPS work to be able to make these engineered organoids to be able to model that in a dish. So this is just a UMAP, again, looking at here's the different cell types that we've looked at in the heart. So we get immune cells, atrioventricular, fibroblasts, progenitor fibroblasts, smooth muscle parasites, endocardial and endothelial. There are different populations. So we can look between the mutant and the wild type mice to be able to see, is there different populations coming up? One is that in the mutant mice, actually, we have more progenitor type of cardiomyocytes. And then in our wild type mice, it seems like we have more of an epicardial profile just based on these cell types. So there is a lot of background to suggest that there is epicardial involvement in this disease. So in order to study the differences between this one aspect of this fatty fibril infiltration, we've been looking at generating iPS cells into cardiac fibroblasts. So there's some good protocols out there now where we can generate cardiac fibroblasts and see differences in fibrosis that may occur. So the mutation might not be causing an increase in fibroblast proliferation, or it might. Or maybe the cardiomyocytes are dying and now there's a space which allows the cardiac fibroblasts to proliferate. So if we really want to look at disease mechanism over time in the development of this pathophysiology, we can using these cardiac fibroblasts. But also another really intriguing, again, aspect of this disease is this fat, this epigenic profile within the myocardium itself. And it always occurs from the epicardium going more endocardium. So the progression of the disease actually starts at the epicardium. So again, what's the origin of this type of fat cells? Is it epicardial derived? Can we model this in a dish? And so we've been looking at generating epicardial cells using our iPS cells, looking at control cells versus ones with mutations in them to be able to say, is it really a difference in the epicardial population that's causing it to be more like a fat cell? Or is it just that there's space and another type of cells coming in? Or is it the cardiomyocytes themselves that are transdifferentiating into into adipocytes, which has actually been proposed as one of the mechanisms. So we've generated epicardial cells, and we're now kind of assessing them not only for their profiles and at the single cell at the monolayer level, but now we're adding different components to the media to be able to say, can we actually push them and to develop fat? And so just our preliminary data, we've only been doing eight days, but we need to go a lot further into this. So if we add different components, we can actually get lipid accumulation in these cells. So we do differences between the control and ACM to see if we can model that. But lastly, we're actually taking our tissue, the EHT platform, and now we're trying to mimic the cellular 3D localization of these epicardials to now coat these different engineered heart tissues with our epicardial cells to see what changes if we do still get the disease modeling of the fat actually infiltrating within the myocardium in this type of cell model system. So this is kind of ongoing projects, and I'm really out of time here, so I'm going to just quickly go through these the conclusions and maybe some caveats that maybe I didn't mention that I think are important. But and I'll start with this last point. I do think the bioengineers really did advance kind of a lot of the problems that people are associating with iPS-derived cardiomyocytes. They would say, well, you know, their sarcomers aren't aligned. And so people would pattern them onto certain substrates and say, look, the centromeres are now aligned. Another problem, well, these iPS cardiomyocytes are immature. And so they would start pacing it, and they would see that they're getting more mature. They would start to express different mature profiles. Well, this they don't have this metabolism differences, so they would add different media components that can drive this. So it really is, within the last 5 years, I think, amazing how far we've come to be able to develop organoids that really do recapitulate a lot of what the heart looks like and how we can use these for even replacing a lot of the animal models out there. And this is just like, depending on your system, depending on your assay that you're trying to address, taking just the platform of the EHT technology type of system, I think is great, but you can always fine tune that and change it. And the students in the lab are really excited in doing these kind of projects because it's something you don't know what will happen, and yet it takes a long time. But once it works, then it's something we set in routine in the lab. And one other great point about these iPS cells is that you do get the isogenic lines. We think this is very important, and that every single paper that's published out there will need those isogenic lines to be able to compare just what that one single mutation difference leads to. Also, these are human cell types. We do see, I've done so much RNA sequencing in my life, there's massive differences between the channels that mice express versus humans express versus the different cells, profiles, gene expression profiles. So having a human cell type is, I think, still more translatable if you could use them for your system than sometimes a lot of people get credit for. And then the last point is just talking about, and what I think a lot of people don't realize is that the mouse it's an entire being. So it has all the organ systems involved. So it has your kidneys, it has the brain. So there's the neurohormonal effects in disease, and a lot of the treatments that we're trying to develop, they're not actually targeting the mutation itself or what the mutation is doing at the primary level. They're actually looking at the compensatory mechanisms. So they have beta adrenergic receptor blockers, they give statins. So that's really not that's the secondary effects that the body is trying to respond to actually heal this defect, and we're now inhibiting that. So iPS cells are great because you can look at now the mechanism of just what that mutation is doing in a closed system. And some people might say, well, that's not what happens in a human, but I think at least it gives us a platform to be able to develop drugs that are specific for the primary early stages of the disease process. So that, I'm just going to thank a lot of my lab that's been working on this. Sobi, who's off in med school now, actually did a lot of that epicardial work. And then we have a few people that are now on the ACM project, and then everybody who's developed this help with the software. My former lab at Stanford, Zachary Lassen's group in Lely, who's been a good sounding board to be able to understand a lot of these disease processes. Collaborators in the Gregorio lab who help us a lot with the contraction or our biochemical, some more of the Western blot analysis. And of course, at Bio5 and the Guter Foundation, which has been incredible support. When you're a junior PI and you're trying to get preliminary data, trying to get fund, the Bio5 has been incredible to be able to say, hey, we like this idea, let's give you some seed funding so that you can get this project up and running. So thank you for that support from the Guters and from the Bio5 Society. And I'll answer any questions you guys have.
Thank you so much, Professor Churko, for your great presentation. I really enjoyed your presentation. Before going to Q&A, I just want to announce, and you take a breath, just want to announce our next speaker. So I share my screen. Okay, so our next speaker is Professor Thomas Gervais. He's the professor in Department of Engineering Physics at Polytechnic of Montreal. He's also the co-founder and CTO at MesoChip. He will be talking about the technology that he developed in his lab on cancer on chip. So if you want to get the most updated information on the upcoming seminars, you can always follow our Twitter account at TransLBME, and also our the co-organizer, myself and Dr. Akbari, on LinkedIn. So we post this, and also I want to thank again Montreal Transmedic, our sponsor, which sponsored this e-seminar from the beginning of this e-seminar series in 2020, June 2020. So by that, I also want to encourage everybody to look at on the bottom of your screen. You see the YouTube channel. There is a bar that guide you to our YouTube channel. You can go and see the previous talks if you missed them. And also, we gradually upload the talks on YouTube channel by permission of our invited speaker. Thank you so much. Okay, so my first question actually coming from basically reviewers who review our grants, and they always ask us, in the absence of, the innervation or immune system in these heart on a chip models, how far we are from the reality, or how far these models are realistic for disease modeling.
Yeah. It's a model. I'm not going to say everything's perfect. If it was perfect, we use it. We would be using the human heart itself. But we're never going to get there if it's not funded. Like, we need that money to be able to develop these different tools to get them to a point where they are perfect. So, and that's just something you really can't explain to reviewers, because they try to find anything that could be wrong. But it's a model system, and there's always going to be something that you can't really respond to, but you could try your best. So you can try to get to a point. If there was an immune component, maybe we can take the buffy coat of our PBMCs, and we're working with other people to do this right now, where we're looking at adding in our own immune cells to these systems. So there's always ways to get around it. It's just trying to convince the reviewer that we need this funding to be able to get to the point where we can actually make the discoveries that we need for using these models.
Exactly. That's the challenge, and we are trying always to convince them, but they don't believe that like these kind of simplified model that we propose usually answer the question, and like how far we are from clinical studies or like clinical outcomes. Anyway, so I think that your answer is really helpful to answer the question for the next round of applications. The other question I had, because you mentioned also about the differentiation of these iPSCs to cardiomyocyte and the heterogeneity of the population. So I understand you said that 90% or up to 100% you can get the pure cardiomyocyte, but in case of 90% cardiomyocyte, how do you enrich them if you want to take out the other cell type that you do? It's not desired in your application.
Yeah. So the great thing about cardiomyocytes is that they utilize different substrates for energy. So you can glucose starve these cardiomyocytes, or the cultures, and the cardiomyocytes survive. They're mitochondrial rich. So if you can actually design the experiment to be able to enrich them based on the physiology of the cell type itself. And if you know that they're mitochondrial rich, you can sort them using mitochondrial dyes. So you can use MitoTracker is one of those to be able to say, now we're just going to purify the cells that have a lot of mitochondria. They utilize lactate. the other cell types don't. So we add lactate to the culture to be able to have the cells, the cardiomyocytes survive, but the rest of them die off. So there are a lot of strategies to get them to the point where they are 100% pure populations.
So now there's a follow-up question that there's a debate in the field. So some researchers think that, for engineering these heart tissues, hESCs, we need pure cardiomyocyte, and some people in Europe or around the world think that we need also to incorporate fibroblasts.
Yes.
And what's your thought on this?
I do think you need fibroblasts. So the fibroblasts, so this is more of a question of making the model itself. If you want to have something that's truly more representative of the heart, you need some fibroblasts, you need endothelial cells, because those help with even just nutrient diffusion. The fibroblasts themselves will secrete different factors that can help the cardiomyocyte. So even in the single cell analysis, you can see that there's cells that will express certain ligands and the other ones that express receptors. So there's communication between these different cell types within the heart themselves. So incorporating these different cell types does make a better model. Like the real question that a reviewer or even someone doing the experiment has to answer is, what can we do to get the answer without spending the entire grant and the entire year? Like, we can make them 3D, we can add every single cell type in there, we can differentiate them and keep them mature for a year in culture, we can pace them. But if you can get the same answer without doing that, then it's better use of research funds to not do that. So yes, we can get them to that point of adding really expensive growth factors, but if that's not necessary, I don't think that's the right way to go about it. But fibroblasts definitely are very good in these engineered heart tissues to be able to make them more sarcomeric alignment, more mature, better, and even epicardial cells now, people are saying they're secreting certain growth factors that are promoting the maturation of these constructs.
Perfect. Yeah, I like the idea also putting these epicardial cells around the cardiomyocyte and your system. I think this is the first time I see such a thing in a dish. And also, like, are you planning to also to start, commercialize this platform in the future, or?
Not really. No, we mostly want to look at the disease itself. We want to look at the disease mechanisms, and we want to be able to develop targeted small molecules or therapeutics for these different. We're not looking to commercialize most of the engineering efforts for this.
And another idea that I like, because you showed us that you built a simulator in the lab, very cheap one, like less than 5,000 or something. And how do you make sure that the output that you get from this simulator is kind of realistic, or like, how do you validate? Because you made it in the lab, and then in terms of engineering, I know that your talented students are electrical engineer or you have collaborators, but how do you validate the output from these simulators?
Yeah, that's a great question, because are we trying to really model the electrical system of the heart? No, it's not the best system, because there's these nodal cells in the heart that are doing this job for us. There's not some current that's being pumped in our bodies all the time, but it's a way that we can activate the cells in a system that's more exogenous, I guess. We can manipulate it outside without relying on a biological cell type to do it, or neurohormonal. So yes, that's actually true, that it's not like very representative. It does work. It does. We do have contractions. The only way that we can, I guess, assess the system itself if it's working is that we look at the beat rate of the contractions versus the frequencies. So we can see that as we increase frequency, we get that increase in beat rate. we try to design this as biphasic so that we don't have ions being built up on one the electrode or not, so that we can constantly go back and forth. But yeah, you can get electrolysis of the media if you're hitting it too hard, and all those other types of comments might go on.
It's cool. It's cool. I really like it. There is a question from, but I don't understand the question. I don't know if you talked about N in your presentation. One of our audience is asking, what is N?
N is the biological replica.
And it's the biological. I don't know. That's why I was confused about the question. And also, I encourage other participants. I know that we have some of our colleagues and researchers who are interested in these talks. In your talk, in audience, if they have question, put their question in the Q&A box. I pass to Mohsen if he has any question.
Let me unmute. Well, thank you very much, Jared. No, I think I really enjoyed your talk. I don't have a lot of any questions at the moment, but one question, or maybe I just have one question about this assay that you when you measure this when you use these two posts to measure the the contractile properties of the tissues. So how thick was the tissue that you formed?
Yeah, it's about 1 millimeter.
Is that in diameter or length? Diameter.
So
did you notice any issues in terms of nutrients, diffusion and
No, no, we haven't. We've in the field. There's actually I don't know if you know Jay Zhang. He's in Birmingham. He's looking at adding in endothelial cells and fibroblasts, and then they can get these constructs up to like 1 centimeter even, and yeah. So it seems like there's not.
So you don't need the vascularization.
Not in this system, no.
Okay, cool.
Okay, and Professor Blaszczak, one of our previous speakers, also asking a question in the chat box. Our information. There is much data concerning N-catering and fibroblast migration and collagen secretion. It would be good to test the effect of N-catering antagonists on cardiac fibroblasts.
For sure, and actually, our epicardial system, we're looking at that as well, because in this disease, we think that there's like an epicardial ENT type of system going on. So we're looking at the different adherents to be able to assess whether that's actually more advanced in our PKP2 mutation or knockout iPS cardiomyocytes. So good point. Thanks. We're definitely looking into that.
And then you can connect with Orest. He's expert in cardiac fibroblasts and in fibroblasts in general. Yes. So thank you so much, Professor Churko, for your great presentation. It was a pleasure to host you today. And I encourage everyone to join us next week, same time, same platform, for the talk from Professor Thomas Gervais on cancer on chip. And thank you so much. Have a great day and take care. Bye-bye.
See you guys. Bye.
Take care. Bye.